Hello, everyone. My name is Jensyn Carr. My partner for this project is Morton, and our mentor is Dr. Ashley Haines. We are doing the first report of microplastics in eastern oysters in the Elizabeth River using the genus and species Chrysostrea virginica. The Elizabeth River is an estuary located in southeastern Virginia that is home to many aquatic organisms, but despite providing a home to these organisms, it is one of the most polluted bodies of water on the U.S. East Coast, and one of the pollutants that may be in the river water is microplastics. Microplastics are plastics that measure 50 micrometers to 5 millimeters in length that are extremely harmful to animals because the animals can ingest them, which negatively impacts their growth and reproductive abilities. Humans may also ingest these microplastics when they consume seafood. And one popular seafood item is the eastern oyster. And again, we use the genus and species Chrysostrea virginica. The eastern oyster is a filter feeding bivalve that cleans the river water by filter feeding. However, as it's doing this, it may filter microplastics back out into the water because previous research has shown that microplastics can be incorporated into oyster tissues.
So due to the incorporation of these microplastics into the oyster's tissue, our overall research goal will be focused around using the oysters as a bioindicator to determine the presence of microplastics in the Elizabeth River. To go further, we will also be identifying the different types of microplastics within these samples. So the sites that we collected data from were the Willoughby Harbor Marina and the Elizabeth River Project Learning Barge. Our methodology is originally based on Thiel et al. And involves chemical digestion, filtering of the oyster tissues, and looking for microplastics within the micrograph. We counted 41 microplastics from 12 micrographs. Preliminary examination of initial samples based on the data shown here do indicate the presence of microplastics. And from Site 2, we collected 208 microplastics from 324 micrographs. These samples shown here do indicate the presence of microplastics.
So here are our Site 1 versus Site 2 comparisons. We have our form averages from Site 1 and Site 2, and in both sites, microfragments were much more common, with more than 80% of the plastics found in these oysters being microfragments. The color averages in Site 1 and Site 2 varied. In Site 1, the oysters ingested a similar amount of pink and blue microplastics, but the oysters in Site 2 overwhelmingly preferred blue microplastics. So there are more microplastics in oysters from Site 1 than Site 2. Microfragments are more common, and blue microplastics are more common than any other color. There was not any evidence of bioaccumulation in larger oysters, and our most conservative calculations suggest that there are more than 1,000 microplastics per oyster in the river.
This being our first report, we were able to prove that microplastics are in the oysters that live within the Elizabeth River and in the surrounding water bodies. This indicates that microplastics are common in the Elizabeth River water. For our future work, we're hoping to collect more samples and use Raman spectroscopy to identify plastic types. In application, this data can be used to work towards developing regulations to guide local industries.
Here are all of our references. And we would like to thank the Inclusive Excellence Grant from Howard Hughes Medical Institute to Norfolk State University, the National Science Foundation STARS Grant, collaborators at the Elizabeth River Project, and Dr. Stephen Vai from the Biology Department. Thank you so much for your time. Again, this was the first report of microplastics in eastern oysters in the Elizabeth River using the genus and species Chrysostrea virginica. My name is Jensyn Carr, my partner is Morton, and Dr. Ashley Haines was our mentor. Again, thank you so much for your time.